Hello students, welcome to Visionary First Step YouTube channel. And in this video, I am decided to take one of the important topic from biological classification. Okay, I explained detailed about the whole topic in one of my video, but I decided to take this particular topic, sexual reproduction in fungi, separately. Because this topic, many students, they feel it difficult to understand when it comes to the concept clarity. So that's why I thought that I would take this topic so that separately if I teach that, students able to understand in what depth they need to understand this topic. Okay, without any further delay, let's get started. Now, sexual reproduction in fungi. When it comes to fungi, we know that it is a multicellular organism. The one exception is Saccharomyces cerevisiae, that is yeast. How they undergo reproduction? In fungi, we commonly observe fragmentation of the hyphae, and that fragmented hyphae, each one give rise to a new fungal body. Another mode in vegetative mode of reproduction is budding. So unicellular organism like yeast, they show budding. Here one cell produces a small outgrowth that develops and it detaches from the parental cell and develop into a new organism, that is budding. Then one more is binary fission. Just like in bacteria, there is one cell that undergo cleavage and two cells are formed. So this is the most common type of reproduction, asexual mode of reproduction in fungus. Then how they undergo asexual mode of reproduction? By producing spores. These are very important concepts. Okay, when it comes to spores, we observe zoospores, ascospores, basidiospores, zygospores, conidia. See, all these are confusing whether they are asexual spores or sexual spores. You need to have a clarity because most of the questions are based on these spores. So remember, conidia, sporangiospore or zoospore, these are actually asexual spores. Okay, now let us take one by one. See here, conidia. Why they are called conidia? Because during the asexual reproduction in fungus, they produce a special type of hyphae that is called conidiophore, which have the ability to produce conidia exogenously. So this is important, whether it is produced endogenously or exogenously, it is important. So where do we observe this particular conidia formation? Okay, we'll try to understand now. Conidia is produced in Ascomycetes as well as Deuteromycetes. Okay, so these are the two types where we observe exogenously produced conidia, and they are asexual spores. Now come to sporangiospores, where they are produced inside the sporangium. That means they are endogenously produced. So there is one classification called Phycomycetes. Phycomycetes. Here we observe sporangiospores. Phycomycetes. So here we observe that there is a asexual spore in Phycomycetes, Ascomycetes and Deuteromycetes. What about Basidiomycetes? In case of Basidiomycetes, spores are absent. Asexual spores are absent. One of the important character you should remember. Okay, in Basidiomycetes, Asexual spores are absent. Absent. Okay, hope you remember this. This is very, very important. Okay, rest of the classifications, they have asexual spores. In case of zoospores, they are motile. Okay, motile spores. I will show you the picture how the zoospores are present. They are commonly observed in the fungus which are aquatic. Now, sexual reproduction is by different types of spores, which includes oospores, ascospores, and basidiospores. Why there is a specific classification in those spores? Because they are produced in the specialized fruiting bodies. Suppose a spore, sexual spore is produced inside the basidium, which is produced on the basidiocarp, then it is called basidiospore. Okay, if it is produced in the ascus, which is produced in the sac-like structure, then it is called ascospores. Similarly, oospores. Oospore is very primitive, means it is not advanced. Okay, so various spores are produced in a distinct structure called fruiting bodies. Now, one of the character is very, very important. What is that? Whether it is asexual spore or sexual spore, they are all haploid. That we should know. Okay, very important. When these spores germinate and produce a hyphae, then the fungal body is also haploid. Then at what stage we observe diploid nature? Is it a very long duration we observe diploid cells in the body, or it is transient? That is very important, and that makes this concept little difficult to understand. Now just observe this. Here fragmentation, can you see this is the hyphae, septate hyphae. When it undergo breakdown, each fragments they grow into a new individual. This is binary fission. Parental cell undergo cleavage and produces the two daughter cells. Okay, then budding, here you can see parental cell produces small outgrowth and that develop into a new one. That is very common in yeast. Now coming to conidia, this you can see here in case of penicillium. This is the conidiophore hyphae which are specialized to produce the asexual spore, that is conidia. These spores are produced exogenously, not inside any sac-like structure. They are produced exogenously. And here you can see in case of rhizopus, this is the structure, sporangium, inside which spores are produced. Here you can see in case of albugo, they are aquatic, they produce zoospores. You know, albugo comes under Phycomycetes. Albugo, rhizopus and mucor, these three come under the Phycomycetes. So whatever given here, it is related to the sexual mode of reproduction, and I am going to discuss in detail about the sexual mode of reproduction, how it takes place. One common step is given. Actually, three common steps are there. One is plasmogamy, followed by karyogamy, and when the karyogamy takes place, zygote is formed. Zygote undergo meiosis. Now we will break down and understand clearly what is plasmogamy. Now there are two compatible hyphae. They come close together and there will be a fusion of the cytoplasm, nothing but the protoplasm. Initially cytoplasm fusion takes place, and that we call it as plasmogamy. Now in that cell, nucleus is separate as it is for a short interval of time, and then it is followed by karyogamy. These two nucleus also fuses. So plasmogamy, fusion of cytoplasm, karyogamy, fusion of nucleus, and when the cytoplasm and the nucleus fused, now cell become diploid. Now that diploid cell undergo meiosis, and meiosis produces the sexual spores. Okay, so one diploid zygote cell produces 4 haploid spores. These are sexual spores. Okay, so this is very common in lower form of fungus. lower form of fungus. Okay, so which are the steps? Plasmogamy, karyogamy, and zygotic meiosis. But there are some exceptions. That is where we are going to focus on. How it takes place, where it takes place, and what is the speciality. Now moving on to next slide. Here same thing I explained, 3 steps. Fusion of protoplasm between 2 motile or non-motile gamete is called plasmogamy, and the fusion of 2 nuclei called karyogamy, and meiosis in zygote result in the haploid spores. So these are the cycle. Okay. Now here in this picture you can observe the produced gametes may be of same size, isogamous, anisogamous, one is large, another one is small, oogamous, one is large non-motile and another one is small motile. It's okay. So this is all about the structures what we observe in the some form of fungus, but not in all. Okay, I will tell you, I will show you the picture. Now when it comes to normal mode of sexual reproduction, where when a fungus reproduces sexually, 2 haploid hyphae compatible mating type come together and fuse. So in some fungi, fusion of 2 haploid cells immediately result in the diploid cells. This is very important. In some fungus, immediately after that nucleus fuses, that we call it as karyogamy. However, in other fungi, Ascomycetes and Basidiomycetes, here there is one intervening stage. That means plasmogamy takes place, but it is not immediately followed by the karyogamy. So what happens? In a cell you observe 2 nucleus, N plus N condition. This is very special in the case of Ascomycetes and Basidiomycetes. This cell having 2 nucleus is called dikaryon, and this phase is called dikaryophase. Okay, so later the parental nuclei fuse and the cell becomes diploid. See, I want to tell you one thing here. Without explanation where they fuse and how they multiply, we don't understand this. That's why in the next slide I try to take some of the information from Basidiomycetes, okay, where they are clearly explained. Okay, so here they have said what is that? Plasmogamy takes place, it is immediately not followed by karyogamy, so the cell is having 2 nucleus and that we call it as dikaryon phase, and later they fuse and karyogamy completes, cell become diploid for a transient time, and that cell undergo meiosis and produces the haploid spores. Okay, this is how the sexual reproduction takes place. Now the reason to take Basidiomycetes is here they have clearly explained what happens actually. Now let us take one diagram for better understanding. Here I will draw. So there are 2 compatible hyphae. As we know, Ascomycetes and Basidiomycetes, they have septate hyphae. Okay, so these 2 are the compatible hyphae. Let us consider these 2 hyphae of the fungus belong to same species. Now cells of these 2 hyphae fuse. Okay, that we call it as plasmogamy. Okay, let us consider here it undergone fusion. Okay, now I will show it like this. Now in this cell there are 2 nucleus because it is immediately not followed by karyogamy. Now this cell goes on divide, and in that case all the cells here produced are having 2 nucleus like this, right? Now this structure produces the fruiting body. Let us consider this is the basidiocarp. This we call it as basidiocarp. Now in basidiocarp all the cells are dikaryon. Remember this, because fusion not at all occurred. but it produced the fruiting body. So all the cells in the basidiocarp and ascocarp, they are all dikaryon, not diploid. Remember this. Okay, dikaryon, single cell having two nucleus. Now here it has basidium. This is important. This is the pouch-like structure, basidium. Inside the basidium, carefully listen. Inside the basidium, now two nucleus undergo fusion, and that we call it as karyogamy. Now for a very short interval of time, transient time, now this cell become diploid and it immediately undergo meiosis and produces the spores. Now here spores are produced. How they are produced? This is also very important. Basidiospores are produced endogenously and ascospores are produced exogenously. Okay, so this character is very, very important again. Now, so this is about the spore production in basidiomycetes. So karyogamy and meiosis takes place in the basidium, producing 4 basidiospores. Basidiospores are produced exogenously. There is one correction here. I think I missed it. In case of basidiomycetes, spores are produced exogenously, and ascomycetes it produced endogenously. Make correct of this. Okay, so I will show you what type of questions can be framed in NEET examination and in what depth you should understand this. See, this is how actually the structure is. Observe, here these two are compatible hyphae. When these two compatible hyphae fuse and form a cell where dikaryon is present, here all the cells produced are again having two nucleus. The same mycelium then produces the fruiting body. Now inside this basidiocarp, again cells are having dikaryon. Now inside the basidium, karyogamy completes, cells become diploid, they form a zygote. Now zygote immediately undergo meiosis and produces the haploid spores inside the basidium. Now these spores are produced exogenously outside the basidium. Let us go with all the statements. Try to solve this question. Select the correct statement from the following. So what is the first statement? When a fungus reproduces sexually, two haploid hyphae of compatible mating type come together and fuse. Now what they can do, they can slightly change the words. Instead of giving when a fungus reproduces sexually, two diploid hyphae come close together and they are incompatible mating type, like that they can make the statements. So you should be very clear that when sexual reproduction takes place, two compatible hyphae of haploid type. So the statement is correct. Okay. Now, in some fungi, the fusion of two haploid cells immediately result in the diploid cells. Is it correct? In some fungi, fusion of two haploid cells immediately results in the diploid cell. This is also correct because in some of the fungus, plasmogamy is immediately followed by karyogamy. Next, fungi, ascomycetes and basidiomycetes, an intervening dikaryotic stage occurs. Such a condition is called dikaryon and a phase is called dikaryophase. This is also correct because specifically in ascomycetes and basidiomycetes we observe dikaryon phase. Now diploid state in ascus, see I am using it very carefully. Diploid state in ascus. Ascus is the structure inside the ascocarp. Basidium is the structure inside the basidiocarp. It is transient. Diploid state is transient because the cell inside the ascus and basidium where karyogamy takes place, zygote formation takes place, but zygote undergo immediate meiosis. That's why it is transient. Now cells in ascocarp and basidiocarp, a fruiting body produced in ascomycetes and basidiomycetes, are dikaryon. This is also correct. Why? Ascocarp and basidiocarp, I already explained. These structures, they are originated from the fusion of two compatible hyphae where plasmogamy take place but karyogamy did not take place. That's why all the cells in basidiocarp and ascocarp are dikaryon, having two nucleus. Now all the statements given are correct. So this is how you should understand how they frame the question. Okay, this is the question framed by myself, but to show you the pattern. Now observe here. Select the incorrect match from the following. Mycelium, when it is produced, when the hyphae, they form a network-like structure, then it is called mycelium. If hyphae is haploid, then mycelium is also haploid. Spores are, I already told you, whether it is asexual spore or sexual spore, they are all haploid. Now basidiocarp, basidium can be diploid for a transient time, but not basidiocarp. Basidiocarp is just like ascocarp, it is also n plus n, not 2n. Okay, so that is why this is incorrect one. Okay, ascocarp is n plus n. Now just look at this question. Find out the correct statement from the following. When diploid hyphae of compatible type come close, plasmogamy takes place in sexual reproduction of the fungus. This is what we discussed, and this is the correct statement. And in sexual reproduction of the fungus, karyogamy is followed by the plasmogamy. Is this statement is correct? Incorrect, because plasmogamy is followed by the karyogamy. So this one is the incorrect statement. What is the next statement? Dikaryon phase is observed in basidiospores and ascospores. This is correct. Karyogamy takes place in Ascus basidium. So these cells are diploid for brief period, which is transient. See how beautifully questions are framed, right? Next. Select the incorrect statement from the following. Asexual spores and sexual spores both are haploid in fungus. Correct. Then sexual and asexual spores both are formed by meiosis in fungus. Correct? Yes or no? Answer is no. Why? Sexual spores are formed by meiosis. Asexual spores are formed by mitosis, because already fungal hyphae is haploid. It cannot undergo meiosis and produce asexual spores. So it has to undergo mitosis. So this statement is incorrect. When spores germinate, they give rise to haploid fungal hyphae. This is correct. In fungi, zygote is usually very short-lived. The zygote immediately undergoes meiosis and produces the haploid spores. As we know, this is also correct. Okay. I hope today in this video I try to make you sure that you will understand the sexual reproduction in fungus in a detailed manner. Okay. So thank you for watching. See you tomorrow.